Assalamu alaikum everyone, my name is Muhammad Abra Zahid and welcome to the third topic of the scary video series, the microarrays. And honestly, this topic is not a very difficult topic to understand. It's just a bit, just really confuses us because हमारे पास ना जो हमारे रिसोर्सेज होती हैं, बुक्स होती हैं, उसमें यूजली इट्स नॉट रिटन दैट वेल, ठीक है. एंड मतलब दे जस्ट गिव इट टू यू टू द पॉइंट. इतना ज़्यादा वो कंसेप्चुअली नहीं लेके जाते हैं. इट्स नॉट अ वेरी हाई यील्ड टॉपिक आइदर, बट इट हैज़ बीन टेस्टेड. ओके, सो ऑब्वियसली इट्स नॉट समथिंग लाइक you could say, for example, like genetic crosses. That is something that's pretty much tested every single year. It's not that high yield, but it is still a pretty important topic and one that I never suggest my students to skip. Okay, so let's get started. Okay guys, I just want you to pause the video and I just want you to read what I have written over here. Okay, let's just read that. Okay, so I hope you guys have done that. Okay, so guys, microarrays, first of all, what I want you guys to know, okay, they're also called DNA and gene chip. So if they've ever used the word gene chip or if they've ever used the word DNA chip, don't be confused, it's the same thing. Okay, so they're basically slides printed with thousands of tiny spots, okay, in defined positions, with each spot containing a known DNA sequence which acts as a gene probe. Okay, let's break all of this down. So over here I've made a very simplified gene chip. Okay, it's a very simplified gene chip. Okay, what I want you guys to really try and focus on is, do you guys see how I have all of these columns and then I have all of these rows, and inside these rows there are these boxes. Okay, so in an actual gene chip and an actual microarray, it's like this obviously is a very oversimplified version. I should write this over here. This is oversimplified, but again, this is the amount of detail that is required to build your concept, and it is the amount of detail that is required in your exams. Okay, so essentially there are like spots like this. So this is one spot, this is the second spot, third spot, fourth spot, whatever. Okay, so that's the first thing, okay, that it's a slide printed with thousands of tiny spots, okay, in defined positions. Okay, so what do I mean by defined positions? Okay, so guys, let me just quickly draw a chessboard over here. Okay, let's just draw it freehand. Okay, all of you have probably played chess, right? So it's been quite some time since I've played chess, so I don't exactly remember what the coordinate system is, but essentially I use a coordinate system, right? A, B, C. Okay, so if I say define this position, this position would be BC or CB. Okay, if I say define this position, it would be DC or CD, right? Like this. Okay, so in that very same way, this entire grid thing, this entire network of spots or boxes or whatever you want to call them, it is it is defined like this. That let's say that this is 1 2 3 4 5 6, this is A B C D E. So if let's say that I have a gene probe over here, so I can say that okay, this is the coordinate 3C. Okay, so that is how it is defined, how it is sort of you could say that it's mapped out. That's a better word for it. So defined position, mapped out positions, and let's move on to why this is important. Okay, with each spot containing a known DNA sequence which acts as a gene probe. Okay, so let's talk about the gene probe thing first. So what is a gene probe? A gene probe, essentially, the word probe means to detect for something, right? That's the definition of the word probe. So if I was to write the definition here, it's essentially going to be the length of DNA that has complementary, just a second, complementary base sequence to another piece of DNA. Piece of DNA which I am trying to detect, which I am detecting, or which I am probing for, checking for. Okay, detecting / probing for. Okay guys, we all know that DNA is it wants to be in a double stranded arrangement, right? Let's say that I am trying to find out whether I have A, C. Let's say that I have a piece of DNA that I am trying to detect. Okay, there's a piece of DNA that I'm trying to detect. Let's say it's A, C, G, T, A, C. Let's not make it too long. Okay, here's my phosphate backbone. Okay, Now this is a piece of DNA that I'm trying to detect. Okay, now what will I do? What will my gene probe look like? So guys, if I make my probe for it, if I make my gene probe for it, what will it be? I want you guys to pause the video and try and make it yourself, otherwise I'll just quickly make it. Okay, it's going to be TGCATG. Okay, same sugar phosphate backbone, and that is what I have. This is the probe. And guys, what will happen? So over here in these spots, I have these gene probes present. Okay, so the piece of DNA that I'm trying to find will essentially come. So the DNA is mobile, the DNA is moving, right? The chip has immobilized. This probe, it's immobilized, it's fixed over here. So the DNA, let's say I have a solution that has different strands of DNA. This strand is just roaming around. This strand will then come and it is going to bind over here. And as we all know, just a quick revision, AT forms 2 bonds, C and G forms 3 bonds. 3 which bonds? Hydrogen bonds. Okay, small things like this you can be tested for. एक से दो मार्क्स से सवाल आ जाते हैं. So guys, this is what's going to be happening over here. Okay, so this is an overview of what the gene probe is sort of like. Okay, so guys, I want to talk a bit more about like gene expression profiling and gene profile, and then let's talk a bit more about what microarrays sort of are like. Let's break microarrays down a bit more. So first, let's talk about these two things. Okay guys, so talking about gene expression profiling, essentially what I want you guys to understand is that not all genes are expressed at a given moment. Okay, let's say that I have a gene for 6 feet 5 inches, but unfortunately I am only 5 feet 11 inches. Okay, so why? Because this gene is not being expressed, right? This gene is not being expressed. That is what is happening. अब वो जिस भी वजह से हो रहा है, वहाँ पे किसी किस्म का एपिस्टेसिस हो रहा है जो उसको इनहिबिट कर रहा है, वहाँ पे let's say कि whatever is happening, I don't care. Okay, right now I don't care. So what I want to focus on is that some genes are expressed and others are not. Okay, and others are not, and that's very important for me, because let's say that I carry a disease, I carry a gene that is specifically coding for a disease, or let's say that I have a disease gene. Okay, that is a gene that doesn't work properly. So I want to check and see that, okay, is this gene active or not? Is this gene active or not? And guys, this is what the microarray tests. Okay, so this is what the microarray is sort of testing. The gene expression profiling is just the study of which gene is being expressed and which gene is not being expressed. It's sort of like a test, you could say. It's sort of like a okay, I'm doing a gene expression profiling test on this person. Okay, that is what is happening. Gene profile, a really simple definition, I've seen it pop up in your exams. So it's just going to be a pattern of gene expression at any one time. So let me just briefly explain this to you. Let's say I am now 20 years old, right? So I have some specific genes active in me. Let's say I'm 45. Now I'm going to have different set of genes being expressed in me. Obviously some will stay the same. For example, hemoglobin will still be expressed in the cells of my hematopoietic stem cells, okay? But still not in my hematopoietic stem cells anyways. Okay, so still, like, you guys get the idea, right? Because some genes, like over here, let's say I have I have some specific gene X being expressed, A being expressed. Over here I have some specific gene X being expressed. So there's a change. So the gene profile is pattern of gene expression at any one time. Okay, so these are just two definitions that I want you guys to sort of make yourself familiar with. Okay guys, so now talking about microarrays. Okay, so what are microarrays really doing? Microarrays are just doing this thing. They are just used in your gene expression profiling, guys. That they're sort of trying to figure out which gene is being expressed and which gene is not being expressed. Okay, and I'll talk more about this towards the end of the video as well. Okay, so now let's move on into let's move back to the microarrays. Alright guys, so now I want to talk a bit more about what's really happening over here. So all of these, different spots, what I want you guys to know, I'm going to write something over here and then I'll explain it to you guys. So each spot can contain, each spot contains the same gene probe, but usually multiple copies of it. What does this mean and why? Let me just write the why so I don't forget that. Let's talk about what this really means. Okay, guys, so what we're going to do is that we're going to take this specific portion. Just give me 1 second. Okay, so let's just take this specific box, okay, and let's zoom in on this box. Okay, let's zoom in and let's make this box right over here. This is sort of like this, right? This is sort of like this. Let's just make it a bit bigger because it will be easier for us to do what we're going to do. Okay, so guys, what I am doing is, let's say that I'm trying to find out if I have the DNA of, let's say, A, C, G, T. Guys, what will be the probe for it? Quickly tell me what will be the probe for it. The probe would be T, G, C, A, right? Okay, so what I'm going to be doing is, let's just copy this, just paste this, let's make it as small as we can. Okay, this is still not fitting, I'll have to write it. Okay, so guys, over here in one of these spots, in one of these boxes, T, G, C, A, and now this will be, just a second, this will be all over here. I'll have to add the T upar wala danda, but you guys see it's the same gene probe that is being repeated over and over and over again. Okay, and there's a reason for that. I want you guys to think of why. Like, why do I have all of these repetitions? Okay, so I have all of these repetitions. Let's just make one last one and now we are done. Okay, so guys, I want you to try and imagine what is really happening over here. Okay, so you see one gene probe, second, third, fourth, fifth, sixth, 7, 8, 9, 10 gene probes in one of these same boxes. Guys, don't you think I could have just kept one gene probe? Why did I have all of these gene probes? Like, what's the reason for that? Let's talk about it. The first is, more likely for DNA to be detected. Guys, let's just think about this in terms of cricket. Okay, let's say that there's let's just even leave cricket. Let's just say there's a catch game. Okay, so there's two scenarios with us. Okay, in the first scenario, there are 10 people that are standing and there's one ball that is coming towards them. Okay, so 10 people and one ball. Okay, and then there's one person and then there's one ball. Okay, let's say this is a circle of radius let's say 40 meters, and the same over here, that it's essentially this person is covering a radius of 40 meters. Okay, like, don't think they're all standing at the same spot, they're sort of spread out. It's a radius of 40 meters, they're spread out. Guys, could you tell me where is it more likely that the ball will be caught? It's a catch game, right? वो जो बॉल फेंकते हैं, कैच जिसे कहते हैं. ठीक है, जब एक बॉल आ रही है और 10 लोग खड़े हुए हैं उसे कैच करने के लिए, या जब एक बॉल आ रही है या और एक बंदा खड़ा है उसे कैच करने के लिए, guys, simple probability says it will be more likely over here. ठीक है, and guys, that is exactly what we do. Instead of having one gene probe over here, we have 10 gene probes. Okay, that DNA का जो piece है, it will be more likely, ज़्यादा मेरे लिए आसान हो जाएगा, ज़्यादा मेरे लिए likely हो जाएगा कि वो आके इसके साथ bind कर जाएगा. ठीक है, बिकॉज इतने ज़्यादा खड़े में एक ना एक के साथ तो बाइंड कर ही जाएगा, ठीक है गाइज. दैट इज़ वॉट इज़ हैपनिंग ओवर हेयर. ओके, दैट इज़ द एग्जैक्ट रीज़न वाई आई हैव more of these. I have multiple copies of this. Okay, the second thing is that it helps in identifying strength of expression. Okay, what does this mean? Strength of expression, guys, I'll do this very briefly, very briefly, but we will be talking more about this once in our next class, where we are talking about how microarrays help us identify gene expression in disease and non-disease cells. But what I want you guys to really know is that, let's say if there's one DNA, let's say यहाँ पे एक DNA का स्ट्रैंड आके बस बाइंड करता है, ठीक है, वर्सेस इन 10 के 10 प्रोब्स पर बाइंड हो जाता है. तो बाय दैट आई नो द स्ट्रेंथ ऑफ माई एक्सप्रेशन, बाय दैट आई नो द स्ट्रेंथ ऑफ माई एक्सप्रेशन. मुझे पता चल जाता है एक्सप्रेशन कितनी ज़्यादा हो रही है, ठीक है. सो गाइज, दैट इज़ वॉट आई रियली हैव. ऑलराइट, so now I want to move on to one last thing, which is sort of this समझ लीजिए कि some essentials about microarrays that I want you guys to know. We've been talking a lot about how microarrays are going to be helping us in determining the expression. So if they ever ask you what is the role of a microarray, the microarray helps to determine expression. It also helps us to determine the strength of expression, or whether expression is being, like if the gene is being expressed at all or not. But we are using a DNA to check this expression. So guys, एक सवाल आता है कि प्रोटीन सिंथेसिस में what is being used? mRNA. So if something has an mRNA, that means it is being expressed. So guys, let's just take the example of my cheek cell. Okay, so guys, my cheek cell probably has let's say what should I say, let's say it has myoglobin. Okay, it has myoglobin being expressed. Myoglobin is being expressed, right? So I will have myoglobin ka, sorry, just a second, I will have myoglobin ka mRNA present, right? mRNA will be present, and the DNA will also be present. लेकिन DNA किधर होगा? DNA होगा मेरे न्यूक्लियस के अंदर. mRNA किधर होगा? मेरे साइटोप्लाज्म के अंदर, ठीक है. Okay, and then let's say that I have a cheek, not a cheek cell, let's go with a cardiac cell. Okay, cardiac cell. Alright guys, so my cardiac cell has, let's say, troponin. Okay, it has a protein called troponin that is being expressed. Same thing that is happening ऊपर, ठीक है. और जल्दी से मैं लिख देता हूँ, mRNA होगा, एक्सप्रेस हो रहा होगा, and also DNA भी obviously present होगा. Guys, I have a very quick question from you. Can you tell me कि cheek cell में troponin की जीन present होगी या नहीं होगी, and cardiac cell में myoglobin की जीन present होगी या नहीं होगी? Guys, just quickly think. They will both be present, because all the cells of my human body are going to have the same genetic makeup, the same genotype. Okay, the same genotype. So guys, अब सवाल आता है कि यार DNA तो इधर भी पड़ा हुआ है, DNA तो इधर भी पड़ा हुआ है, तो फिर how can I use my microarray to determine my expression? तो guys, अब मेरी बात सुनो, ठीक है, मेरी बात सुनो. हम कर क्या रहे होते हैं कि microarrays, man, this will become more clear to you guys once once you also talk about the entire technique of microarrays in the next video. But in microarrays, what we are doing is we're taking mRNA, we're converting that mRNA to DNA, and then we are using the gene probe to test for it essentially. Okay, quick revision. mRNA to DNA कैसे जाते हैं? We go through the process of reverse transcription. Okay, don't forget that. All right, and enzyme कौन सा होता है? Reverse transcriptase. So this is a very simplified version. I completely understand if you guys don't get this अभी, ठीक है, because इस पे भी मैंने पूरी वीडियो करनी है, but guys इसको इस तरह अंडरस्टैंड करने की कोशिश करो. अगर मैंने देखने की अच्छा चीक सेल के अंदर मायोग्लोबिन प्रेजेंट है तो मैं क्या करूँगा, मैं मायोग्लोबिन का mRNA निकालूंगा, ठीक है, या मैं जो भी mRNA है सेल के अंदर मैं उसको निकालूंगा, मैं उसको DNA में कन्वर्ट करवाऊंगा, एंड देन आई विल सी कि ओके मेरे पास माइक्रो एरे के अंदर, ठीक है, इज दिस पॉजिटिव है या नेगेटिव है, वट एवर इज हैपनिंग. कार्डियक सेल में क्या है, कार्डियक सेल में आई विल बी टेकिंग द mRNA. गाइस मुझे एक चीज़ बताओ, अगर मैं चीक सेल का DNA लूँ, इफ आई यूज द चीक सेल्स DNA, विल दैट गिव मी a proper microarray result or not? It will not. Why? Because microarray is determining the expression. It is not determining whether the gene is present or not. It is not determining that. It is just telling me if the gene is being expressed or not. Okay, so cardiac cell, if I take this DNA and I use the DNA to do the microarray, nope, wrong, because the DNA will include the DNA will include genes for proteins that are not being expressed. For example, keratin will never be expressed in a cardiac cell, right? So हमारे पास कार्डियक सेल में DNA में केराटिन पड़ा हुआ होगा, ठीक है, केराटिन की जीन पड़ी हुई होगी, लेकिन वो एक्सप्रेस नहीं हो रहा होगा. तो माइक्रो एरे के लिए हम हमेशा mRNA इस्तेमाल करते हैं. सो इफ दे एवर आस्क यू, इफ दे एवर गिव यू अ क्वेश्चन स्टेम दैट ओके हम लोगों ने क्या किया, हम लोगों ने सेल लिया है, उसका हमने न्यूक्लियस पियर्स किया है, न्यूक्लियस में से DNA निकाला, उस DNA का हमने माइक्रो एरे करवा दिया, विल दैट गिव मी अ करेक्ट माइक्रो एरे रिजल्ट और नॉट? इट विल नेवर गिव यू द करेक्ट माइक्रो एरे रिजल्ट. ओके, सो कीप दैट थिंग इन माइंड. Alright, so that wraps up our video, and I just want to write something over here, all over the place. I know this, guys, all over the place. ठीक है, अभी आप लोग थोड़े से कंफ्यूज हो गए, थोड़ा सा दिमाग घूम रहा होगा. प्लीज़ वेट टिल द नेक्स्ट वीडियो, ठीक है, बिकॉज दिस टॉपिक इट्स जस्ट वन ऑफ दोज टॉपिक्स, बिकॉज ये हमने कभी पहले पढ़ा नहीं होता. बाकी चैप्टर्स आप फिर भी कह सकते हो, जैसे क्रोमोसोम्स थे, ठीक है, वी नो अबाउट दिस, वी एटलीस्ट नो अ बिट अबाउट दिस. ओवर हियर इट्स अ कंप्लीट न्यू जोन, इट्स अ कंप्लीट न्यू एरिया, ठीक है, new territory. So it will take some time. Watch the next video, and then trust me, everything will come together, every single thing will be coming together. Okay, so guys, that's all. Thank you so much for watching the video. I hope you guys enjoyed, and please do watch the previous videos as well, and like and subscribe, and thank you so much. Allah Hafiz.